Hi, my name is Alan Katzenmeyer. I'm the Chief of the Aquatic Ecology and Invasive Species Branch at ERDC. Today we're at the Aquatic and Wetland Ecosystem Research and Development Center. I'm going to show you around the facility and some of the work we have going on for endangered species conservation and invasive species management.
Hey, my name's Ian Knight. I'm a research biologist with the Environmental Lab, and I work on the biological control of invasive aquatic weeds. At the moment, I am working on the alligator weed flea beetle, which is a biological control agent of alligator weed. Alligator weed was introduced to the US in the 1800s and was actually part of a really successful biological control program in the 1960s and 70s. But what wound up happening was over the subsequent decades, alligator weed has started to move northward into more temperate climates and has become an invasive species in temperate climates where the tropical beetles have failed to establish. So the work I'm doing at the moment is investigating cold tolerance among different populations of beetles that have already been released into the US and comparing them with populations from South America, along with some collaborators with USDA ARS. And hopefully we'll be able to identify populations of flea beetles that are putatively more cold tolerant and more suitable for biological control in these temperate climates.
Hey, I'm Audrey Harrison, and I'm a research entomologist here at ERDC. I'm working on a macroinvertebrate colonization study in the lower Mississippi River. Aquatic macroinvertebrates are the invertebrates that live underwater, and the Mississippi River is full of them. And with this project, which is a colonization study of macroinvertebrates, we are looking at different natural and artificial riverine substrates and seeing what types of macroinvertebrates are associated with different substrate types or habitat types underwater. The importance of doing that is to be able to figure out which habitats hold the most biological diversity, which habitats we want to protect, enhance, create more of, or conserve. And in doing so, we have a way to quantify the benefit of various restoration projects in the lower Mississippi River.
Today I'm going to be talking to you about some of the research we're doing on bighead carp. What we're really focused on in this lab right now is how carp interact with increased salinity. So we're exposing them to an acute salinity experiment where we greatly increase salinity over a matter of minutes, and a chronic salinity experiment where we slowly dose them with higher and higher amounts of salinity over a 1 to 2 week period. The reason we're interested in this is because as we operate flood control structures and during regular flood events, sometimes we create freshwater bridges out in the Mississippi River, and we want to make sure those bridges don't create new pathways for those carp to spread. Invasive species really impact our native ecosystems by pretty much outcompeting our native species. Native species are going to have natural predators, whereas an introduced species may not. Therefore, it can grow and double much faster than the native species, effectively outcompeting them for food and resources.